Good day to everyone. I am Beltricia Sara Elangido, Fabria Lupli Bernardo, and John Vergi Agabao are with me today to present our research titled Cytotoxicity and Phytochemical Screening of Mimosa pudicaline, Makahiya leaves using branch lethality assay test BSLA as potential cancer cells inhibitor. But before we begin, let's define cancer. Cancer is a disease in which certain cells in the body grow uncontrollably and spread to the other parts of the body. Cancer is one of the leading causes of death worldwide. There is a study on Makahiya leaves, Mimosa pudicaline, that it has pharmaceutical potentials due to the presence of phytochemicals like alkaloids, flavonoids, saponins, and terpenes. Its great microbial activity against Staphylococcus aureus and Bacillus subtilis, and the antioxidant property it exhibited. Cancer treatments including surgery, radiation, and chemotherapy is painful or stressful for patients.
The material used in the experiment.
In preparation of plant powder, first is 200 grams of Mimosa pudicaline or Makahiya leaves were gathered. The second is the fresh Makahiya was washed with water and sun-dried for 3 days. After drying, the leaves were pounded with mortar and pestle. Subsequently, the researchers collected 50 grams of powdered Makahiya and two setups was made. Control setup that doesn't have powdered Makahiya leaves and the experimental setup with the powder. And in the hatching of brine shrimp, were put in the test tubes with artificial seawater. Artificial seawater was prepared by dissolving with 38 grams sea salt in 1 liter of tap water for the hatching of brine shrimp's eggs. When the now placed in the test tube with artificial seawater mixed with powdered plant covered under a lamp to serve as an incubator for 24 hours, the number of surviving shrimps were counted manually and tabulated. The procedure done 3 times.
The preparation of plant powder and the preparation of brine shrimp lethality assay. Table 1 Phytochemical Screening. Table 2 Number of surviving brine shrimp. The result of Table 1 shows that Mimosa pudicaline contains a chemical components that can inhibit the growth or development of actively dividing cells. Alkaloids, flavonoids, saponins, and terpenes was shown its presence the phytochemical testing. The result of Table 2 shows that the Makahiya powder has a significant effect killing the surviving brine shrimps. Analysis implies also that the Makahiya powder leaves are positive for cytotoxicity based on the number of brine shrimp that survive. Approximately 82% of mortality rate was computed.
It is highly recommended to test powdered Makahiya leaves to determine the chemical components of the plant that can inhibit the growth or development of actively dividing cells. Furthermore, it is suggested that the Makahiya powder will be used in different concentrations. In addition, it is suggested that it can be used in the other plants as a component of some medicine used to treat cancer or tumors. It is also recommended that the future researchers find appropriate counting techniques for the experiment.